So here we are, Friday, the first of our 2 virtual days. With any luck, we'll all be back in person on Tuesday. I can do this without my mask on, so that's not entirely a bad thing. And today what we're going to start talking about and going over is genetic engineering, which is, I think, really kind of the coolest thing about our understanding of the science and where we are today. So, why don't we get started? Hit the present thing, do the screen. It's going to go kind of weird with the infinite tunnel of me, which I kind of love. That won't surprise anyone. All right. Genetic engineering. All right, so we've gone over how DNA works and how the letters are turned into proteins, and we understand that the proteins are what actually does the stuff and gives us the traits that we have. Once we really understood this and we had looked at everything, we came to the realization that all living things share the same DNA, the same structure. That means there's no requirement that a gene stays in one species. You can take a gene from any species, put it into the genetic code of a different one, and that new creature, organism, whatever, will still be able to utilize that DNA to make the proteins that the gene says it should make. So we can start to alter the genetic code around a purpose, around a goal. Now, what you see on screen here is a tobacco plant that was genetically modified and spliced with a deep sea jellyfish. So the deep sea jellyfish does a bioluminescence in the dark. Now that plant does the bioluminescence too, because the gene doesn't matter what organism it's in. Everything reads the A's, the T's, the C's, and the G's the same. So we've used this for other things too. This rabbit, it doesn't actually glow. What it does is it produces a phosphorus compound in its fur. So when you shine a black light at it, it glows very brightly. We've done this with mice as well. And of course, once again, you can tell the ones that were successful because once you put the black light on them, they glow that phosphorus green. Now, this was trials. We wanted to be able to prove that we could do it. And this was a way for us to visibly see that we were either successful or not. But since then, man, we have started to genetically modify almost everything. You can look at the list here. Mostly we genetically modify crops. Why? Crops and plants in general are not thinking, feeling organisms. So it doesn't bother us morally if we screw around with their DNA and try to mix things up, add things. And if it fails, and the plant dies. Yeah. We can see some really bizarre examples of this. Now, this is not actually an example of genetic engineering. This is one that I use as an example of what it could do. These are literally just apples that have been soaked in grape juice, so they pick up some of the flavor. I had a student a couple of years back actually order a dozen of these. They were not cheap either, just because after seeing it, he decided, I got to try that. Not impressive, but we could genetically engineer an apple to produce grape sugars. And then biting into an apple would be very much like biting into a crunchy grape. There go the lights, because if I'm not moving, every 15 minutes. All right. Now, we have used this more importantly to try and take care of some major diseases like diabetes. So when I was a kid, 1982, you can see their diabetes supplies cost so much. A lot of people were dying simply because they could not afford them in any way. Now, for most people, this is not true for everyone, obviously, with the, advent of auto pumps and things like that, prices can vary, but a standard testing kit and injection supplies costs a little under $100 a month. As of 2012, when I got this data. And the reason why is because we learned how to genetically modify a bacteria. So what we did here, and let me get my pointer on. All right, so what we did was we took a section of human DNA and we found the human gene for insulin, right? And we cut that out. Then we took a bacteria. We took out its DNA and we cut that open. We inserted, you can see the purple right here, we inserted the human insulin gene into the bacterial DNA, put the bacterial DNA back into a bacterium to let it survive and read its DNA, let it grow and do its thing. And in reading that human insulin gene, the bacteria now produce insulin. Now, before, we would need to kill pigs, excuse me, and we would drain their blood and we would squeeze out their pancreas in order to get the insulin. There'd be a lot of fluid, a lot of mass, a lot of waste, obviously, and then you'd have to purify the whole thing in order to get a dose of insulin. It usually took about 2 pigs to get one human's month of insulin. Well, now we just have these gigantic vats just full of these bacteria, just going. And when we want to get insulin, we open up a tap, we pour them out, we cook it so the bacteria die, we pass it through a coffee filter effectively, and what you're left with is water and insulin. Simple, easy. This has saved so many lives by this genetic modification. Now, despite that, genetic engineering continues to be a very debated topic. On the good side, all the stuff that we've talked about, the ability to feed the planet, the ability to manage disease. Right now, they are working on pigs, like you can see there, who have been genetically modified so that they do not produce protein tags on their tissues, on their cells. Right now, the biggest challenge for organ replacement, let's say your liver starts to fail, right? We can't always just get a new liver. Well, if they're successful with these pigs, we will be able to just take a pig liver, connect it into you. Your immune system will not fight back because it won't have any tags on it that say, I'm a pig. And theoretically, we could have an infinite supply of replacement organs. Now, on the downside, the bad side there, you can see a lot of people kind of fear what this will lead to. How far are we going to push it? Jurassic Park, the original movie, and then everyone after that was largely a criticism against the science of genetic engineering, with it being dangerous and unknown and having effects that people could not really predict. That said, it hasn't stopped us. We've been genetically modifying organisms now for decades. Agriculture has by far been the biggest beneficiary of this. You can take a look here at all the different genes that we have added to different crops, and this has led to more debate over it. Genetically modified organisms right now, a lot of people are kind of wary of. There is zero scientific evidence that there is anything harmful in any GMO. People are concerned at the materials and the DNA that's now been included into them and what happens if it gets into us. But you have to remember, we've been eating foods our entire lives. All of those foods have always contained DNA. None of that DNA somehow finds a way to incorporate itself into our cells. We break it down. We break the polymers into monomers, and then we use those to rebuild. So any fear that the DNA will somehow affect us is just wildly impossible. That cannot happen. A better concern would be, all right, we add in a gene to produce so that the crops produce their own pesticides inside their tissue. Is that dangerous to us? Well, if it was, we wouldn't have genetically engineered it to make that. But that's a more valid fear that some people have. If we take a look agriculturally, you can see there's just the amount of crops that we have that are not natural anymore are massive. We rely on it. Without it, we could not feed the population that this country has, let alone the population around the world. Naturally occurring crops would require so much more land and therefore so many more farmers that it would not be feasible, not with the amount of land that we have as farmland now, not with the amount of people we have doing farming now. If you want to check this out and really read into it, it is available in the slideshow. Please feel free to take a couple minutes to look at it or pause the video. All right, so to follow that up, if we look in Schoology, right, I've got our virtual Friday folder right here. And if you open this up, here is the slideshow that I just did. This is a video from Kurzgesagt. Once again, watch it. It's fantastic. It explains all of this GMO stuff on the good side and the bad side. And let me talk about our genetic engineering lab. Now, this lab is for our lab class. So that is Bell 2-3 today. On Monday, this will be reposted for the afternoon class, Bells 7 and 8. But for the genetic engineering lab, it's pretty straightforward. Here is what it looks like. All right, our goal to understand the methods and the goals of genetic engineering for whatever purpose and the process. What are you going to do? Well, here's your procedure. There's an 84-letter sequence of DNA on the last page. There's also a list of 15 new genes. You're going to decide on 3 new genes to include, and you have to find the right place to put them in. And then it says remake the final genetic code. I'll show you. It's very, very simple. So we're going to determine a species, whatever species you want. You're going to decide what your goal is. What are you modifying them for? You're going to pick your 3 genes out of the list. Then this is the tricky part, right? Then you have to check the DNA and you have to find the right place to be able to enter it. Allow me to show you. All right. So here are all of the genes. I'll come back to this in just a moment. Here's the worksheet. So let's say I decide I am going to modify a turtle, like soup. What is my goal? Well, I get to choose. Do I want it to be aesthetic, just to change its appearance? Agricultural, personal, which really personal just means that it's something other than the other 3. Or commercial, I want to sell it. I'm going to go personal. Why? My goal for this organism is it's personal first. Why? I want to create a world-destroying turtle. Godzilla will be jealous. Okay, that's my goal. Now, what genes do I want to include? Well, this is where we come up here, right? Here are the genes on the left. They all have these fancy gene names. In the middle is the effect that it has. And then on the right hand is the DNA that it requires. Now, in order to put a new gene in, we must find the right place. I have color-coded these in red. So in order to find the place for IR3, if we wanted to do that, we would need to find the ATC section immediately followed by a CCG. But we'll worry about that in a moment. All right, what do I want? World-destroying turtle. All right, I definitely want HGHA1 because that's going to make him bigger. No problem. So I want HGHA1. I'm also going to want, let's see. I will want it to grow I'm going to do LMA2 so that it's toxic. LMA2. And finally, let's see. You know what? I'll go with GHA9 just to make it poisonous all around. GHA9. Right? So those are the 3 genes I'm going to do. Don't worry about this one. That's not going to help us so much just yet. All right, so let's take a look at our first one. HGHA1. HGHA1 requires me to find ATC, TAT. And that's where I'm going to cut the DNA to put this new gene in. So ATC, TAT. So I got to come down here now to the whole thing, and I need to find an ATC, TAT. So right here is ATC, but it's not followed by TAT. Here's another one, no TAT. There's another one, nope. Nope. Here it is. There's ATC, TAT. So I found the right place. Now what I'm going to do is I want to cut between it. That's where I'd cut. And then this is where I'm going to include the other DNA. So HGHA1 will include GTGCAT. GTGCAT. So I am telling the lab effectively that what I'm going to do is I'm going to cut between those 2 codons, and I'm going to include these 2 new codons in order to give it that new larger size trait. Then you want to come back up, pick your next one, LMA2. Find it. It's here. So here is the combo that I'm going to need. I'm going to need ATC, TTA. You would come down. You would try to find that, draw another line, include the other letters. Hopefully, you guys get the idea. It's pretty straightforward. You're only going to do this with 3 different genes. That's it. Just 3. And then you're done. There's no additional questions. We're just practicing the process of genetically modifying one organism to do something different. So I hope that's pretty easy. It should be simple. There should be, I hope, nothing confusing about it. And I hope this helps. Once you're done with this, there is indeed a turn-in right here. So if you do this online, feel free to turn it in. If you do this on paper, because you can print it, you can bring it in when I see you on Tuesday. If you need to, you can also literally just write this out on a sheet of paper. You can write out this last page right here. What is the organism? What is the goal? What are the 3 genes? And then write out the DNA. It's a little bit more work, but that's how it is. And then you can turn that in. So any of those will work. I hope that helps everybody to better understand it. When I see you, we'll go in greater detail. We'll talk more about some of the really cool examples of genetic engineering and where it's going now. Ladies and gentlemen, I hope that you have a great day. How do I stop recording now? There it is. All right, folks, have a great one. I will see you again on Monday, where we'll do the same thing all over again.